Okay? So I'm very glad to have Chang here to give a talk on the long read mapping against the pangenome graph. Chang is a PhD student at University of California, Santa Cruz, and I'm very eager to watch her talk here. Thanks again for being here, and please go on with your talk.
Okay, yeah, thanks for the invitation to come speak today. I'm Xian. I'm a grad student in Benedict Payton's lab at UCSC. Um, and I'm part of the VG team. So, VG is a big, unwieldy toolkit of lots of different pangenome tools. So, we can do things like construct a graph, manipulate a graph, find subgraphs, get graph statistics. And we can also do some kinds of analysis. So we can map reads. We've got three different mappers, and then we can also do downstream analyses like genotyping variants and doing RNA analysis. But my primary project has been VG Giraffe, which is one of our three mappers in VG. This is the most recent one, the most, I think, multi-purpose mapper. We published this a couple years ago, and we showed that it was very fast, pretty accurate, and was pretty good with variant calling. Um, but it was really designed for short reads and wasn't great with longer reads. So the algorithm for the original short read giraffe was very targeted to short, highly accurate reads. And when we applied it to longer reads, which are very long and very error prone, it wasn't great. It was super slow, used a lot of memory, and kind of killed our computers. So there are existing long read mappers that do work. So I think GraphAligner and MiniGraph are probably the most well known. There's also GraphChainer and Paneliner, which use components of GraphAligner and MiniGraph respectively. But each of these also has their own limitations. So GraphAligner tends to be really slow, particularly as the graph gets more complicated. And MiniGraph was designed to really only align reads to graphs constructed with MiniGraph. So MiniGraph graphs don't include smaller variants. Um, so it's limited in that it can only really align to very simple graphs. So we wanted to make a new long read mapper that would be both fast and generalizable to more complicated graphs. And since we already had a short read mapper, we started with adapting that. So in VG, we work in the variation graph model. So we have a pangenome, which is basically just a collection of haplotypes. And from those haplotypes, we use a graph construction method to get a variation graph out of it. And the variation graph model incorporates both the graph itself, so these nodes and edges, as well as the haplotypes. So the original haplotypes in the pangenome can be represented as paths through the graph. And these are the file formats that you might see if you use VG. So anything ending in G basically is a representation of the variation graph itself. Um, and then the GBWT, which stands for Graph Burrows Wheeler Transform, represents the full set of haplotypes. And I threw in a bunch of citations throughout the presentation if anyone's interested in background reading. Okay, so this is the input to our giraffe algorithm. So we've got a read up here, which I'm representing as a bunch of colored blocks that correspond to where they would align in the variation graph. So the original giraffe algorithm follows a pretty standard mapping framework that pretty much all mappers use. So seed, cluster, extend. Uh, Everyone uses it pretty much. So the first step of seeding, we want to find very short alignments between the read and our reference. So we call these seed alignments. They're just short matches. Um, and for the original giraffe algorithm, we used a minimizer index, which is basically a dictionary mapping a k-mer sequence from onto the haplotypes of the reference pangenome. So pretty simple. We didn't change this in the long read algorithm. The next step is clustering. So we take all of those seeds that are scattered throughout the reference and we try to partition them into groups of seeds that are close to each other on the reference pangenome. And since distance is kind of a hard problem in a graph context, we use this distance index to figure out how far away the seeds are from each other. And this is a useful step because it helps us focus in on which regions of the genome the read is more likely to map to. So if you've got a big cluster with lots of seeds, it's more likely that the read goes there than somewhere else where there's fewer seeds. And then in the original giraffe algorithm, we take those seeds and we go to the haplotypes to do alignment. So within a single seed in the cluster, we extend the alignment as far as we can along the haplotypes without allowing any gaps. And this is the step that really made the original giraffe algorithm very fast for short reads, because we expect that the reads are short and pretty much error-free, or at least they don't have a lot of indel errors. And we also expect the haplotypes to cover most common variants that we find in the human population. So because of this, we would expect the read to match exactly at least one of the haplotypes in the graph, meaning that we can find the entire length of the read at this gapless extension step. If we don't find it at gapless extension, we'd go on to continue the alignment in graph space, which is a much slower and memory-intensive process. And so this is why the short read algorithm didn't really work for long reads, because as we see here, it's less likely that the entire read is going to match exactly one haplotype. So we do a lot less gapless extension relative to the length of the read. And we spend almost all of our time trying to align the read to the graph itself, which really exploded the memory use of the algorithm. So for our long read algorithm, we went back and changed our clustering step, where we just find a big mass of seeds, to a chaining step. So chaining is also a pretty standard algorithm for long read mapping. The goal is to find an ordered set of seeds that are collinear in the read and the reference. So that means that they occur in the same order in the read and the reference. So in these little alignment blocks, none of them overlap. You can walk from one to the other in the same order in both the read and the reference. And another thing we found was that the distance index that we used to calculate minimum distance wasn't fast enough for chaining. So we introduced a new data structure called a zip code tree that I'll talk about more later. So now that we've got a chain of seeds, this forms the backbone of an alignment. And now we have to do alignment. So pretty standard. We take the seeds from the chain, project them onto the haplotypes. And then we do alignment in between the seeds and out from the ends of the seeds. And we try to make this as haplotype consistent as possible. So again, we start trying to do alignment along the haplotypes until we hit a point where they're no longer haplotype consistent. So here in this example, these two alignments can't reach each other on any of the haplotypes. So we go back to graph space and do alignment there. Yeah, and doing that, we can get the full alignment. So this is an outline of the final long range giraffe algorithm, pretty standard. And then the data structures that we need to support all of the queries we make. And if you ever actually run Giraffe, you might notice that there's a lot of indexes that we require. We get a lot of complaints about that, but this is why we need lots of indexes to make it fast. And these are all the different file formats that we require. So the GBZ contains both the haplotypes and the graph, but you also need the minimizers, the distance index, and now this new zip code thing. Right, so for this presentation, I'm going to be focusing on the chaining step because it's the part that I worked on, and I think it's also the most novel of all of these steps. Right, so chaining, again, is a pretty standard problem. We're given a set of seed alignments between a read and a reference, and we want to narrow that down to an ordered set that are collinear in the read and the reference, so none of these overlaps. In a linear context, this is usually found using dynamic programming. So this is an algorithmic technique to kind of break down a problem into subproblems. For chaining, the idea is you want to find the optimal chaining score of a chain that ends at a particular seed. So if we order these, we want to find the optimal chaining score for this seed. But as we go through the algorithm, we assume that we've already found the optimal chaining score for all previous seeds. So we found the chaining score for the best chain that ends at this seed, this seed, this seed, this seed. Right. So this is the dynamic programming recursion for Minimap2, which I've cited up here. They're generally pretty similar, so I just used this one as a common example. So what this formula means is the chaining score for any seed I as we step through the algorithm is going to be the maximum of this formula for all previous seeds j. So we check each one of these individually. And the actual formula for the score here, this is the chaining score for seed j plus the number of matches between I and j. So we look at the sequence between the seeds in the read and in the reference and count up the number of matches. And we subtract from that the gap cost between I and j. So the same thing, but we look for the gaps. The gap cost definition can vary between different algorithms, but in general, both the gap cost and the matches depend on the distance between the two seeds in the read and the reference. So in a linear context, that's pretty easy to calculate because we know the offset of each of the seeds in the read and in the graph, in the reference. But in a graph context, that becomes a much more difficult problem because distance and reachability are both not very well defined in a graph. So as an example, between this orange seed and this purple seed, they're not reachable in the graph, so we can't order them and we can't really find a distance. And more problematically, we often see cycles in a graph, in a variation graph. And those are really not easy to deal with in a dynamic programming context because when we want to find the distance from one of these green seeds to the other, the distance isn't well defined because of this loop here. We can take this loop an arbitrary number of times and just keep increasing the distance between the two seeds. So there has been some previous work done on chaining in a graph. So these two, Graph Chainer and Paneliner, are two mappers that have used their own chaining algorithm. One of them is from Gerald Jane. He's got a whole series of papers that are really great on chaining. If you read them, I would recommend reading them in order because I didn't at first and it was quite difficult to follow. But I think both of these chaining algorithms are based on this original one for sparse dynamic programming in DAGs, DAGs being direct acyclic graphs, so it doesn't include these cycles here. But the basic idea here is that we take a path cover of the graph. So we find like linear paths and then use those linear paths to predict or to estimate distances in the graph. So if you're interested in this subject, this is a great place to start. But for giraffe, we already have an idea of minimum distance that we've been using. So we used that as a starting point for our training algorithm. And to understand the distances that we used, I'm going to have to go into a little bit of background. So to start out, I'm going to introduce the snarl decomposition. So snarls are a method of finding organization in graphs because graphs can get really big and complicated. Snarls help us simplify them and think about them in more distinct pieces. So formally, a snarl is a subgraph of the graph that's disconnected from the graph or connected to the graph by two boundary nodes. More conceptually, you can think of a snarl as a site of variation. So in this purple snarl here, you can take either of these paths through the snarl, representing two different variants in your original genome. And then the two boundary nodes represent common sequences. So this here is a snarl representing two different variants. Oops. This one here is a snarl representing a duplication. So as you walk through this graph, you can take the duplication edge and walk through the node multiple times. And snarls generally occur consecutively along the pangenome, and we call a sequence of nodes and snarls that share a boundary node a chain. And we describe the organization of snarls and chains in the graph using a snarl tree or snarl decomposition. So here, this gray chain containing everything has two child snarls, this purple one here and this green one here. And then snarls can also be more deeply nested than that. So for example, in this snarl, it has two children. So there's kind of two paths you can take, right? There's this node here, and then you can take this path, which in turn contains another snarl. So this is how we break down the graph into smaller and smaller pieces that we can describe using a snarl tree. And for completeness, this is what the full snarl tree would look like. Each node in the graph is the child of a chain, and sometimes the only child of a chain. So this purple node is its own chain. But for the sake of this presentation and keeping things on a single slide, I'm just going to represent it like this. So the other useful property of the snarl decomposition is that we can kind of eliminate some of the complexity of the graph by picturing a snarl as only containing its child chains. So from the perspective of this purple snarl here, even though there's all this nested variation down here, we can imagine that it only contains two very simple children, and then it becomes this simple bubble. And because of this property that snarls are separable, they're only connected through their two boundary nodes. Doing this doesn't change the topology of the graph, right? It's still this pretty simple bubble that it would be like this. Right, so that's this property here. Snarls and chains are only reachable through their boundary nodes, and this property is useful for calculating distances because when we look for the distance, the minimum distance at least, between two positions on the graph, the minimum distance from anything within a snarl can be broken to anything outside of the snarl can be broken up into the minimum distance from within the snarl to the boundary of the snarl and the minimum distance from the boundary of the snarl to the second position. And that's because there's only 2 points through which you can exit the snarl. So technically you could also go this way, but there isn't a path there. Okay, so that property is the basis of our snarl distance index. So this is what we used in the original short-read giraffe algorithm for calculating distances. The idea is that we break up a minimum distance calculation along the snarl tree. So to go from here to here, we use an index to find the distance from every point along or every snarl and chain to the boundary of its parent. So from the position to the boundary of the snarl, from the boundary of the snarl to the boundary of the chain, and so on, walking up the snarl tree for each position, and then we find the distance between the children of the common ancestor. And in this example, because I'm limited to the size of a slide, it doesn't look like this is a big win. Um, but if you look at an actual graph, this snarl-based distance metric is its complexity is linear in the depth of the snarl tree, which tends to be pretty shallow, versus something like Dijkstra's, where the complexity is based on the size of the graph. So this is much faster. In practice, it's constant time. But when we used it for chaining, we found that it wasn't fast enough. This is uh, really a problem with computers because the problem is really all the memory accesses, because we stored a huge index for each snarl and chain, and we had to go look up stuff in that big index every time. So we've created this new, smaller data structure called a zip code um, for calculating distances between positions. So we store one zip code for each node, and the zip code contains a minimal amount of information to be able to calculate distances between two zip codes. So it stores the path to reach it in the snarl tree, so chain, snarl, where it is in the snarl tree. And then for each snarl and chain, it contains the distance index, the distance information you'd need to calculate distances. So like the distance from the snarl to the end of the chain, the distance from the chain to the boundaries of its parent snarl, etc. Um, so finally, getting back to chaining, we use those zip codes to create this zip code tree. And the goal is to be able to quickly calculate distances between seeds in a graph, with the goal of chaining. Um, and ideally, since we want to make giraffe generalizable, we want to be able to handle duplications and inversions and all that topological complexity that we see in graphs. So the intuition behind zip code trees is that we want to make a subgraph of the original graph with the seed positions such that we can calculate distances between the seeds in a traversal of the subgraph. And in order for it to be used with chaining, with the dynamic programming algorithm, it should be a directed acyclic graph, or a DAG, which means that there's no cycles, none of that weird complexity that we tend to see in real variation graphs. So to create this zip code tree structure, the input is seed positions on a graph, and each of the seeds has a zip code associated with it, so we can quickly find distances. And we're going to use the snarl tree kind of as a guide for creating the zip code tree. So first, we make one node in the zip code tree for each seed in the graph. So pretty simple. Each seed here is a node in the zip code tree. And then we add a node for the start and end bound of every snarl and chain that contains a seed. So this chain is the big root chain containing everything. We add its start and end bound. Then we do that for the child snarls here. So the start of the snarl, the end of the snarl here and here. And we do that walking down the snarl tree for everything containing a seed. And technically there'd be another chain here, but I ran out of space. Okay, the next step is to order the snarls, the children of snarls. So in a graph, these children of the snarl are unordered, but we assign it an order in the zip code tree. And when we can, this is a topological ordering of the snarl children. When the snarls are dags, um, when they're not, we just kind of guess at a topological ordering. And then once we have that ordering of the children, we can add in edges. So the edges occur between nodes that are adjacent in the graph or in the snarl tree. So between children of the chain, we add in an edge. And then between children of the snarl, we add an edge. And for snarls, since we've ordered the children in a topological ordering, we add an edge from each child to every child that came before it in the topological ordering. So again, for a DAG, that would capture the full connectivity of the snarl because it's in a topological ordering. But if the snarl isn't a DAG, so in this case, it's the snarl isn't acyclic. You've got this looping edge here, and just adding edges like this won't capture that looping edge. And we want, as I said before, we want these zip code trees to be acyclic because it makes everything easier. But we also wanted to capture the topology of this graph. So to sort of include that duplication edge, we include information from the read. So we allow these nodes that break the ordering of the read to split up into two separate children. So even though in the original graph, these two seeds came from a single node, in the zip code tree, we allow them to split up into separate children of the snarl. And then we order those separate children based on the ordering in the read. So this one comes first, and then we add edges as we would for a normal snarl. So this is the final structure of the zip code tree. For the sake of the presentation, again, I'm simplifying it. The topology is pretty much the same. I just took out the some of the chain boundaries. And then in the actual data structure, we store the zip code tree as this one linear vector of nodes and edges. And this improves our memory colocality. So, when you're looking things up in memory, it's faster to look things up that are close together in memory versus far away. And so when we go to find distances in a traversal of the graph, everything we're looking for is pretty close together in this linear representation. So yeah, we find distances in a traversal of the graph, and we add up the distances on the edges to find the distance between seeds. And in doing this, we can use these distances for the purpose of chaining. So now if we've got these seed alignments between the read and the graph, or this zip code tree, we can find the best chain of seeds by walking through the zip code tree and finding the graph distances from the zip code tree. And this would be the ordering of seeds from the distances in the zip code tree. And the nice thing about these zip code trees is that even though they're acyclic, they're pretty simple. There's no loops or inversions or anything. We can find this path here, which in the original graph corresponds to this looping path here, which we wouldn't have been able to find using the original graph because we can't really deal with loops like that. So that's how zip code trees deal with duplications. We can also deal with inversions, again, by taking information from the read. So in this graph, this motif here represents an inversion. You can traverse this node going forwards, or you can take these inversion edges and traverse it going backwards. So using the information from the read about orientation, we can tell that we need to traverse this seed going that way. And then when we go to make the zip code tree, we can make sure that we include the seed in the correct orientation. Right, so that's how we do chaining. We use the zip code tree for distances. And because the zip code tree is created for a set of seeds, we do zip code tree construction once per read. So that adds a new step to the algorithm. So now it's seeding, create the zip code tree for calculating distances, and then we use the zip code tree for chaining, and then we do alignment. And alignment isn't really a new topic, so I'm not really going to go into it. But if you're interested, these are the graph aligners that we use in Giraffe. So we've very recently merged our long range giraffe algorithm into the main branch of our GitHub repo, meaning that it'll be available in the next release of VG, which should be soon, I think. And running it is pretty easy. We keep the same interface, but we now have a new option for running long reads. We just use a different parameter preset. And then we've also added this new index, the zip codes. So that can be built with VG auto index like normal. And so here are some preliminary results. This is showing the runtime and memory use of different long read aligners on hi-fi and nanopore reads. There's a bunch of different graphs and aligners, so I think we should focus in on this one here. It's the most comparable. This is it's kind of poorly named. The HPRC graph is actually this whole mess here. So what it's saying is it's from the Human Pangenome Reference Consortium version 1.1 of all of the data constructed with mini-graph cactus using CHM13 as the base reference genome. And the D9 means that we've frequency filtered the variants. So out of the 90 or so haplotypes, any variant has to be in at least 9 of them to be included in the graph or something like that. So it just takes out the very uncommon variants to take out a lot of the complexity. And for short read giraffe, if you're not doing haplotype sampling, this is the graph we recommend using. So yeah, here I'm comparing giraffe and graph aligner, mapping to to that graph. And the conclusion is that giraffe, is much, much faster than graph aligner. So it's actually faster than this graph would make it seem because we had problems running with graph aligner. It took more than like 5 days, which is the limit for running things on our server. So it got kicked off, and I had to rerun it with twice the number of threads. So to make this a fair comparison, I think the graph aligner numbers should be doubled or so. But in any case, Giraffe is much faster than graph aligner. We do use quite a bit of memory, and again, to be fair, the memory used for graph aligner should probably be halved. But yeah, we're still comparable to the other mappers. In terms of accuracy, we're pretty accurate here. I'm showing rock plots on 1,000,000 simulated reads. And again, there's lots of different conditions here. So I'll just highlight giraffe and graph aligner against the same graph, and then mini map 2 mapped against CHM13. And we're more accurate in terms of just the number of reads mapped correctly. And we also have better map keeping quality calibration. So mapping quality is supposed to be a measurement of how confident we are that the read got mapped to the correct place. Mapping quality of 60 means that we expect there to be a 1 in a million chance that the read was incorrectly mapped. So for 1,000,000 simulated reads, we would expect to get one incorrect map U60. We actually get 150 and 1,500, which is not great, but it's still better than graph aligner and minimap 2. So good accuracy, better mapping quality calibration than the competition, but still not great. And simulated read accuracy is not like the most robust measurement of actual performance. So we also did variant calling. So here I'm showing small variant calling with deep variant, and then structural variant genotyping with VG call. And then for the small variants, I'm comparing against Minimap 2 in orange. And then for structural variants, I'm comparing to GraphAligner in purple. And here I'm just showing the F1 value as like an overall measurement of how we're doing with variant calling. And in general, the blue points representing giraffe are doing better than the other points, um, except for indel calling on hi-fi reads. Minimap 2 is doing better than us, but we're not too worried about this because for short reads, when we ran deep variant, we trained a model to specifically use giraffe reads, and we saw a really big improvement with the trained model. But for long reads, we haven't done that yet. This is just with the default model, which is trained on Minimap 2. So we expect that when we train a model, we'll improve. So not too worried about this gap. And yeah, that's all I have. So Giraffe, we think is in a place where we're ready to share it with the public. It'll be in the next VG release, but we haven't published yet. We're still waiting on some new results. There's going to be a new set of HPRC graphs released soon, so we want to test Giraffe on those graphs to make sure that it's robust to the bigger and more complicated graphs with more genomes in them. For short reads, we now recommend haplotype sampling as the recommended pipeline. But it doesn't work as well as we expect it to with long reads, so we're still debugging that a bit. And the long read code path with chaining doesn't work on paired end reads yet. We still use the old code path for that. So just for the sake of keeping our code clean, we're planning on extending long read giraffe to work with paired end short reads. And then we need to publish, and I need to graduate and get a job. So that's it. Thanks to my whole lab, everyone in this picture, and particularly the people highlighted who are working on this project with me. And thanks for listening, and I'll take any questions.
Thank you very much for your talk. It was very nice and very clear. We already have some questions from the audience. So I'm reading the first question. Thanks for the very interesting talk. To better understand the results, do you know more or less the dimensions of the graph, number of nodes, number of beings, and so on? Apologize if I missed those data.
Oh, number of nodes and edges, I don't know. It's I think 92 haplotypes though. Um, yeah, I don't have a good sense of the node
Okay.
count.
Okay. And another question is, what is the definition of correct alignment in the preliminary
Oh,
results?
yeah, that's a good question. So when we simulate reads, we label the reads with where they come from in the or we simulate from a graph containing the um, the sample we want to simulate from, and then also the reference. And then using that graph, we can project the reference coordinates from the graph onto um, the simulated reads based on the graph alignment. So all of our reads have graph coordinates, or sorry, reference coordinates of where we expect them to map in the reference genome. And then we can do the same thing when we map them to our actual pangenome. We can project the reads onto the, or we can project the reference coordinates onto the reads to see where they overlap with the reference. And then we use those reference coordinates to calculate accuracy. So anytime the read has any reference coordinate that's correct anywhere in the read, we count it as correct. So it's a little bit permissive in that anytime the read overlaps anywhere with the correct placement in the reference, it's counted as correct.
Okay. And how do you simulate the reads?
Yeah, so VG has its own read simulator. I don't know how exactly it works, but we create a graph containing the reference genome and then the sample we want, and then we simulate from the sample.
And do you have any idea of the maximum error rate that you are able to? engine?
Oh, that's a good question. I'm not sure. I think we've tested it on D9 or sorry, R9 breeds. And it worked. It wasn't great, but yeah, we haven't tested it on like ancient DNA or something with a lot of errors. But I imagine that as we add more and more errors, it's just gonna use more memory. So it depends a lot on how much memory you have available.
So you think it's more a matter of the memory that you need?
Yeah.
I see. I see. Thank you. Let's see if there is any more question from the audience. Wait a little bit, but okay. So it does not look like there is any more questions. So thanks again. I really liked your talk. It was very, very nice, very clear, and so I wish you well for your job hunt.
Thank you.
Okay.